Hello, good afternoon and good morning, everyone, and thank you for joining our Avantor webinar today. My name is Maria Castaño, and I am Life Science Global Campaign Manager at Avantor. I'm very pleased to welcome you all to this webinar about getting started with nanopore sequencing. What you are missing matters. Next slide, please. I'm glad to introduce our speakers to you. Aaron Pomerant, Segment Marketing Manager, and Frederick Kohler, that is Marketing Manager from Oxford Nanopore Technologies. Next slide, please. In this webinar, you will learn about how real-time nanopore sequencing works and how researchers are using the technology to uncover novel biology. Experimental design, including sequencing DNA, RNA, and methylation, targeted sequencing options, and analysis workflows. How to get fast and accurate results, and latest customer case studies, including microbiology and infectious disease research. Next slide, please. And before I hand over to our speakers, I would like to provide some important information about how the webinar will progress. After the presentation, there will be a Q&A session. Please submit your questions in the questions chat. They will be answered in the Q&A session and directly by mail if we would run out of time. Our guest from Oxford Nanopore will handle and answer the coming questions. This webinar will be recorded, and you will get the link by email after the session, so you and your teams can go through it at your convenience and when you need it. At the end of the webinar, you will also receive a survey, and we would appreciate if you could please fill it. It's very important for us to get your feedback to improve the quality of our webinars. And finally, for additional comments or questions, you can always contact us on webinar@avantorsciences.com or visit our website at bwr.com. Thank you all and enjoy the presentation. Frederick, please welcome and you can start.
Thank you so much. So welcome everybody. So I'm going to present a little bit more on how nanopore sequencing working. So let me first start by telling you a little bit about the company. Next slide, please. So Oxford Nanopore is bringing a new generation of molecular sensing technology. So mostly our first use is our DNA sequencing. But next to that, we are looking further with RNA sequencing, and probably in the future we can do also protein sequencing. With this idea in mind, we wanted to make it available for anyone, anywhere. Therefore, it needed to be affordable, easy to use, and accessible. We also wanted to have real-time data for rapid insights, and this all in a scalable device. So we have portable instruments to ultra-high output. In the field and in production lab can be used. So what you're missing matters, so meaning that there's important information in your DNA. Therefore, it's needed to have the ability to read any read lengths, as well native DNA as PCR. Because we're looking at native DNA and RNA, we can also have methylation information in there. And of course, we're looking at all genetic variations like SNPs, structural variations, and assembly, phasing, and as said, full methylation. So let's go to the next slide. So at Oxford Nanopore, we are offering an end-to-end platform. So this means that you can have library prep, sequencing, and data analysis. We have kits that can be used in the field and in the lab, which have simple workflows and can be automated. Also, our devices can be used in the field and in the lab, from low to high output platforms. And analysis can be done quite simple till also making your own analysis systems. Next slide. So how does it work? So if you look at the DNA and RNA, it goes through the pore, and this pore is embedded in a membrane. So in the library prep, we add a motor enzyme to the DNA. And with this motor enzyme, it will make sure that it fits to the pore and does a single strand through the pore. This all goes on a speed of 400 base pairs per second. Through the membrane, we are putting an electric current, and this electric current will change if the DNA comes through. And the signal that you then get will we can be put a base caller in that. So what this features? So you can do direct sequencing of DNA and RNA, getting a really rich information. There's no PCR needed, so no amplicon bias. As said, we can do read lengths, doesn't matter what kinds, and it will be in real time. So after the first minute, you will have some first results. And it's scalable because we're using the same technology in the different platforms. You don't have any problem moving to a bigger platform. Going to the next slide. So as already said, we can read any read length. Depending on the kit that you are using, you can have normal read lengths, which are at a minimum of 20 base pairs to ultra long 4 million base pair. This can be done with our ultra long kit. This can be needed for if you're looking at de novo assembly. Next to that, as already said, methylation is included because we're looking at the native DNA and we're looking at what the voltage change. This is giving us that extra information that can be needed for your project. Next slide. So with this, we can actually do any kind of sequencing. So if you're looking at genotype, targeted or not, we can look at whole genomes, low pass or standard, full assembly, and even T2T. It's giving you all the possibilities to look at SNPs, methylation, structural variation in the way that you need it. Next slide. So we're working together with Avantor in a nice partnership. And with Avantor, we are selling the MinION. Next to the MinION, we are having the different flow cells, the MinION flow cell and the Flongo flow cells, but also the different kit. And coming soon, also the GridION. Next slide. On the next slide, we can look a little bit to the different flow cells. So the Flongo is a disposable flow cell, which is low cost and can be used for small genomes, so plasmids, viruses, and bacterial sequencing. It will give you an output from around 1 gigs. If you want more throughput and more gigabytes of data, it's better to go for a MinION flow cell. Here you can look at multiplexing small genomes or even a low pass sequence of a larger genome. And this will give you more data. Next slide. We already talked about the PCR-free DNA libraries, so just let me explain the difference between the libraries that we are bringing to the market. So we have ligation-based sequencing and ligation-free, which is based on transposome. So if you're looking for the ligation-based sequencing, these are really for optimizing and best output and accuracy. So it's all singleplex, and you will need some third-party reagents. Overall, this will take you around an hour to 2 hours to prepare your library, and next to that is possible to barcode your samples. For the transposome-based sequencing, you can use the rapid sequencing kit. This is a quicker library prep, so within 10 to 15 minutes, you will have your library prep done. So if speed is important and you want to get started soon, this is the kit to do it. Of course, we also have this with barcodes. Next slide. Next to the ligation kits and the rapid kit, we can also do targeted based, and it's targeted based can be done in different kind of ways. So we have our adaptive sampling, which is unique for Oxford Nanopore. So basically what we do, you give a file to your Minnow, our program, and in this file you tell actually which genes of interest you want to sequence. So your DNA will come to the pore, and if it comes to the pore and it will read it for 1 second, so 400 base pairs, and it makes a decision by aligning this to your file. If it is in your file and the gene of interest is red, then it will go through. If the gene of interest is not in your file, if this gene is not in your file, then it will eject. Be aware, this can also, of course, be used in a different way and turning it around. So think about host depletion. This can also be used. Next to that, we have some targeted PCR-based kit as well. One of the things that we have is our 16S kit. So this is an amplicon based for the full length of the 16S gene. It has an end-to-end workflow, and we'll come back to that later. Next slide. So overall, these are all the kits that we have. So we have DNA based, and this can help you to select the kit that you need. So if you look at DNA, we have the ligation sequencing kit and the rapid sequencing kit and the ultra long. Both are all already mentioned before. Next to that, we have the barcoding kit and the 16S kit. But we also have RNA kits, and this can be the direct RNA kit and the cDNA kit. Here you have a nice overview that you can get via your Aventor account manager to help you choose what is the best kit for you. Next slide. So with all of this, we can give you insight in all kind of investigations. If you want to do an assembly, single cell, fusion transcription, all these kind of things can be done with nanopore sequencing using the technologies as whole genome or whole transcript or targeted or metagenomics. This all will bring you, next slide please, to give you a full insight of what is there. So generating ultra-rich data for the answer with impact. Now we will move to my colleague, Aaron.
Thank you so much, Frederique. That was a fantastic introduction to the technology. So now that we transition, I think that was a really nice setup into what the technology is capable of. So we learned about some of the rich insight that we can obtain by sequencing native molecules, really of any read length. And moreover, there's quite a lot of flexibility in what you can do with the platform for specific applications. My colleague Frederique mentioned that through our partnership with Avantor, we offer the Flongle flow cells and the MinION flow cells, either on the MinION instruments or coming soon on the GridION. So we're really excited about that offering for yourselves in the community. So with that, we'll talk about some of the main applications for the remainder of the talk. Many of these are along the lines of synthetic biology applications, microbiology, and infectious disease, which fit really nicely again on that MinION or Flongle flow cell application. There we go. So let's take a step back and talk a little bit about some of the applications in this space and where the technology adds a strong value. So my colleague had mentioned some of the accessibility features of the platform. So at Oxford Nanopore, we really try to make genomics accessible. It's really our goal to make it so the technology is not high capex, but can be implemented on site in just about any laboratory in the world. And even off-world. In fact, we have instruments that are on the International Space Station today. So again, they're quite flexible and can go where no sequencing has gone before. Real-time data can be quite critical, especially if you're working in time-sensitive cases like infectious disease, where you really want to get a quick answer. And so historically, genomics has been a barrier, in part because you maybe needed to ship your sample somewhere else to a core facility, and this can add to delayed turnaround times. So again, not only the ability to have more of a decentralized model, but also the ability to generate data in real time can be quite impactful. So what we see in the middle column here, for example, is the fact that with nanopore data, you can stream your live data to your computer, and then you can start to look at that information even after minutes of sequencing. And then lastly, we'll talk about some of the features that are quite advantageous for long-read sequencing. So we're an agnostic platform, but of course there's a lot of value in getting long stretches of DNA sequencing done. And so we see an example here where it's quite impactful for closing genome assemblies. And this is an example with bacterial genomes that we'll touch on more in a moment. So we believe it's time for a replacement of some of the old way of doing things. So of course, Sanger sequencing technology has been around for several decades now, but we see quite a lot of adoption with nanopore sequencing to add more value to what you're trying to understand, especially with your synthetic constructs. So we see examples here where the technology is being used for whole plasmid verification, not just sequencing the small insert. We see it being used for general amplicon sequencing and genotyping. Again, you're no longer limited by short lengths of PCR. If you amplify something that's 1,000 base pairs, you can sequence it on the platform. If you have a longer amplicon that's 5 kb, you too can sequence that on the platform. We'll talk about bacterial 16S and whole genome sequencing in some more detail. But again, we get long read lengths that give you more taxonomic information and the ability to detect multiple microbes within your sample. And then lastly, for anyone who's working in biopharma or cell and gene therapy space, we see quite a lot of adoption, again, to verify not only the plasmid constructs, but also viral vectors that are quite important for gene therapy, such as adeno-associated virus. And again, the ability to sequence the entire construct, not just a partial region of it. So let's talk for a moment about plasmid verification. So as we all know, if you're working in a molecular biology lab, this is really a cornerstone of any molecular biotechnology lab today. So plasmids, of course, are fundamental research tools in basic research, but also for industrial applications such as industrial protein production. As we just mentioned, they can be used in gene therapy, as well as mRNA and vaccine development, as well as genetic engineering. So, but there's been, I would say, a bottleneck for many years when we actually verify these plasmids. So, of course, a routine thing that can be done is you have primers, you can submit them to a core laboratory, and perhaps they would use Sanger sequencing to check an insert in the region. And so that's quite effective for just the region that you're looking at, but there could be a lot more going on with that synthetic plasmid construct. So there could be mutations in the backbone, there could be dimers, there could be concatemers. These are all things that can be missed if you're really not checking the entirety of the construct. So as we just learned about with our rapid barcoding kit, this is really trivial now to just QC the entire region of that synthetic construct. So this can also work across a wide range of different complexities. They can be GC rich, they can have different features like repetitive elements. This is no problem when you're using nanopore sequencing. So not only can you quickly and for a low cost get more information, you can also really rest assured that you're verifying the integrity and the sequence identity before you move on to your downstream experiments. So we see quite a lot of excitement in the community adopting these kind of tools. You can see some examples here in the community for whole plasmid verification, how it's been truly transformative for their cloning pipeline. The ability to really replace a lot of orthogonal tests. So you might be able to avoid, multiple digests, colony PCRs. If you wanted to, do primer walking, that adds quite a bit of cost and complexity. So here you really have a single use case where you can use our end-to-end plasmid verification, again, through your Avantor partner, and an end-to-end workflow. So this gives you a brief overview of what that workflow would go about. So as we just saw, we offer rapid barcoding kits. So really everything upstream of that is up to you. Your general approach to picking colonies and doing plasmid preps. And again, we have recommendations, especially through our partners with Avantor, of kits that we suggest for plasmid preps that work quite well with nanopore sequencing. So that can all be done with those library prep kits. And then when you're ready, when you have your DNA ready, you can use the rapid barcoding kit. So as we just saw, this will introduce a random cleavage in the DNA molecule, and that adds our barcodes. So then you can pool all of your samples, and then you can add our motor protein, and then you're off to the races. You're ready to start sequencing. So that whole process for library prep can be completed for up to 96 samples in less than an hour. Here we see an example of the analysis pipeline. So once you've generated your data, we also offer solutions where you can analyze and get a report out. So generally speaking, you would generate your base called files to your local system or computer. And then we have a program called Epitome, which is a suite of software tools that are really point and click, meaning you can just upload your FASTQ files and it will do the rest for specific applications. So we see an example here of this clone validation report. On the left-hand side, you see an interface of what that would look like. It produces an HTML report, which gives you quality metrics, and it also does quite a lot of analyses for you that you don't need to do any bioinformatics on. So it's all done in the background once you've installed the software. And it's quite nice because it will produce a closed assembly, and it will also annotate all the features of that construct and give you that in an intuitive graphical interface, as well as text files and the files to your laptop as well. So we see an example that actually was an Avantor webinar we hosted a little while back with a customer at Vertex Pharmaceuticals, and you can see their quote here where they've really called out the fact that this is a very simplified strategy and interpretable output, which allowed them to run this in their workflow for assessing their plasmid constructs for gene therapy. Excellent. So moving on a little bit, let's talk about some of the general microbiology and infectious disease applications with Oxford Nanopore. So if you're working in a laboratory and you're doing any experiments that involve, let's say, culturing of microorganisms, this is quite an exciting workflow because you basically, for a low cost and a simplified workflow, can generate the entire genomes of your bacterial isolate that you're working with. This can be used for infectious applications like outbreak detection, antimicrobial resistance monitoring, or really just general basic biology if you really want to get a closed, perfect genome. This could be an application for you. Full-length 16S, this again is an area that's exciting because when you're using this very high parallel sequencing approach, you can generate data to detect polymicrobial detections, but also whole microbiomes. And the fact that we use a long-read sequencing approach for full-length 16S gives us higher taxonomic resolution, oftentimes at the species level. We also have solutions for targeted viral whole genome sequencing. This has been used quite a lot for things like SARS-CoV-2 whole genome to look at emerging strains, as well as targeted antimicrobial resistance tests such as tuberculosis drug resistance. And then finally, we'll touch on whole metagenomic sequencing, which again gives you the ability to avoid PCR altogether and really look agnostically what's in that sample if you're trying to detect a pathogen or any other type of microbe. So just as a brief bit of background, for those who might be interested in epidemiology or pathogen surveillance for infectious disease, this has really been used with the nanopore technology since it was first released in 2014. So it started out as actually one of the early use cases where the public health community was able to take these sequencers out into regions that occurred near or at the site of an outbreak. So for example, Ebola in 2015, 2016 was one of the first use cases for this sort of field deployable genomic approach, which was able to provide data much faster. South America, we see examples with yellow fever and Zika and many of these emerging zoonotic outbreaks, where again, it's quite important to have genomic data to understand the strain, if it's novel, and also what's emerging. And then, of course, in light of the COVID-19 pandemic, many labs around the world had scaled up their sequencing infrastructure to build up that capacity for genomics. And so today we see more labs than ever are using nanopore sequencing for many of these applications. And so at Oxford Nanopore, we try to provide end-to-end solutions to make that process easier. So what we see here, for example, is one of our first end-to-end applications for microbiology. And so this is what we call our no-miss protocol. So this stands for Nanopore Only Microbial Isolate Sequencing Solution, or no-miss. Effectively, this is taking a pure cultured isolate, picking that colony, and doing whole genome sequencing on that sample. So again, what we try to offer is the ability to generate very complete and high-quality microbial genomes, and not just that, the ability to resolve their mobile genetic elements such as plasmids, locate where antimicrobial genes could be located, and again, a system that's very flexible. So you don't need a very high throughput laboratory This can also be used in low to medium throughput labs as well, depending on how many samples and throughput you have. So what we see here is a little more information on what you get out of sequencing microbial samples with nanopore. So when you're using short-read sequencing technologies, you can gather quite a bit of information from that genome, but it's quite difficult to get a complete genome assembly. So we see an example here. It's called a bandage plot, and you can see it's quite a mishmash of these contigs that are generated. So a resistance gene, as indicated in that line, is somewhere within that assembly, but it's quite difficult to understand where it is because short-read sequencing is unable to generate complete enclosed genomes. On the other hand, what we see on the right-hand side is the same sample where we've used nanopore long reads. So we've assembled the complete and circular bacterial chromosome, as well as the plasmids. And so we can find exactly where that resistance gene is located. And again, this can be quite impactful for healthcare settings and distinguishing plasmid transmission from, say, if it's on a bacterial chromosome. So this gives us a lot more rich information on understanding these microbial genomes. We're also, as you heard earlier, able to read out not just the DNA itself, but detect methylation. And so that's quite interesting for microbial genomes. So in bacteria, as with all living organisms, they have not only A's, T's, G's, and C's, but modified bases. So these are often due to methylation patterns. And in bacteria, the common methylation patterns are 6-methyl-A, 4-methyl-C, and 5-methyl-C. And our base caller can actually read that. We've used information from microbial genomes to actually improve our accuracy even further because we can more easily distinguish, say, an adenine from a 6-methyl adenine. So this is now putting nanopore technology in a position where we're generating very high Q scores. So really what this means is historically you may have had to polish with another technology, but what we see now is Q scores. These are quality scores for genomes for microbial samples that are above Q50 and oftentimes above Q60. What this really means is if you had one microbial genome that was 5 million bases long, you would have maybe less than 5 errors across that 5 million base pair genome. So that's excellent. That means that you can really use this as a standalone tool to give you high resolution data, such as calling SNPs or for outbreak detection. And that's quite nice from a cost savings perspective and a time saving perspective. So this again is an overview of what that whole genome sequencing of microbial isolates workflow would look like. So you can see here we would start from an isolate in a culture, perform cell lysis, DNA purification. In our community protocols, we have recommendations for DNA extraction methods, and this can depend a little bit on the microbial sample. Is it a gram positive, a gram negative? Is it a tough to lyse organism like Mycobacterium or yeast and fungi? We have recommendations for those types of samples. So we can drop it in the chat here as well during the webinar, but we can offer links and resources depending on your microorganisms of interest. For this, we also recommend the rapid barcoding kit, which we saw for the plasmid workflow as well, and then again, an end-to-end solution. So this is the analysis pipeline. So just as you saw for clone validation, we offer this for bacterial whole genome assemblies. So it will assemble the entire genome and then perform a number of downstream steps. For example, we can annotate the genes, we can look for resistance profiles, and we can do typing as well. Again, all generated in an intuitive graphical interface report. So again, this might be of interest to you if you're interested in microbial genome assemblies. You really want to get high quality data for a low cost and quick turnaround time. Looking at typing if you're really interested in just quick identification of a microorganism, and perhaps you want to look at more of the antimicrobial resistance profiling. And in the food safety context, we also have rapid workflows for Salmonella serotyping and other microorganism typing as well. So it's quite versatile, and again, a workflow that we're quite excited to see what the community does. I'll now move on and talk about more of a targeted approach. As my colleague highlighted, this is an end-to-end kit that we offer for 16S sequencing. So again, this might be of interest to you if you're trying to understand what is the microbial community that I'm looking at. So is this something, again, in basic research, environmental sampling, maybe even in industry and biomanufacturing, really trying to understand if there are bacterial contaminants, for example. So for this, you can use the 16S kit. What we're showing here is if you look at the 16S rRNA gene, you'll know that it's a long region, and it has hypervariable regions within that gene. Now, classically, if you're using short-read 16S sequencing, it's limited by the read length. And generally speaking, that will only allow you to capture maybe one or two of those hypervariable sites within that gene. Effectively, this limits your ability for taxonomic assignment. So if we say look at Sanger sequencing or short-read sequencing, the ability to get species level resolution is often unattainable. You might get just a genus level characterization. As we know with Sanger, if there are multiple species within the sample, that can be a challenge. Short-read sequencing can also verify if there are multiple species or at least genera within that sample. But with nanopore technology, you get the best of both worlds. So you get high resolution data. You can look at single samples if they're isolates or entire microbial communities or microbiomes. And again, the ability for doing this with a rapid library prep kit at a low cost and quick turnaround time. So here we see an example of what the kit itself looks like. This was shown just a few moments ago, but this gives us a little more information on how the kit works. So again, you would extract your DNA sample, and then you would prepare this with our PCR-based full-length 16S kit. It's quite simple to use, in part because the way that we add our chemistry, our barcodes, and our motor protein are all added on during the PCR step. So it's very hands-off. You effectively run PCR, and then you get your up to 24 samples per reaction. And of course, as you're getting the theme of as well, we have a solution for that on the analysis side. So for this, again, you can plug in your data. Again, whether it's through our 16S kit, or maybe you've done other amplicon sequencing, like for example, ITS for fungal sequencing or 18S, you can upload this data to our Epitome tool, and this will do analysis for you. So it's also quite flexible. You can pick the database that you want to look at with your samples, and your mapping strategy. So maybe it's Kraken2 or Minimap. We offer tunability for how you want to run your analysis workflow. But again, this is all done without any command line, no bioinformatics necessary. It's all a nice point and click intuitive report. And as an output, you get a number, again, of these plots. For example, some of these taxonomic assignment plots and alpha and beta diversity. So it's quite a nice tool for an end-to-end workflow. Here we see one last case study when we're looking at how the community is using and benchmarking the performance of 16S with nanopore. So I'll walk you through it just in a moment. We see, for example, that again, our data quality is excellent. So 99% on a raw read level accuracy is quite exciting for 16S because again, this means that really with a single read, you can capture species level information. So what we see in the plot, the bar plot on the right-hand side, this is a figure from a recent publication where they're benchmarking the accuracy at species level for microbial community profiling. So all the way on the right-hand side, you see two gray plots. This was when they were trying to call species level using short read sequencing. And effectively what you can see here is that you can't really call species levels when you're using that short read information. Again, you can confirm genus level most of the time. In the two middle columns, we see Oxford nanopore sequencing compared to another long read sequencing platform. And effectively, they're equivalent in terms of the microbial community profiling. So we see, full-length 16S really is the way of the future. Its cost is reduced, and it gives you much more information down to the species level many times for that high-level taxonomic profiling. Okay, and then we'll touch for a moment on some of these targeted approaches that can be used for viral whole genome sequencing, or really any kind of targeted approach. So generally what will happen is if labs are interested in doing viral whole genome sequencing, they might be a laboratory, maybe they're an infectious disease lab or a public health laboratory, and they're interested in getting more information beyond, say, a PCR result that would only tell you a binary result of yes or no. Are you positive or negative for detection of this microbe? So generally speaking, once samples are detected and it's a known positive, they can be sequenced. And so generally what we'll do is split that into two PCR steps. These would contain overlapping PCR primers. And then we can use our barcoding kits. This could be the native barcoding kit. This could be the rapid barcoding kit if you have longer amplicons. Of about 1 kb is our recommendation if you want to use the rapid barcoding kit with amplicon sequencing. And then finally, we of course have an analysis tool that will help to map your data to a reference genome and call variants. And this is a very effective way and a quick and low cost way of screening for variants within the community. It's quite a flexible workflow. So what we see is an example here over the years where really this methodology has been adapted to just about any viral genome that you can think of. So of course, SARS-CoV-2 whole genome sequencing. The example that we're showing here is the Arctic approach. This was using 400 base pair amplicons across the genome. And you can see, for example, if your amplicons are short, then you need a lot of amplicons to cover the full length of that genome. So you can see that 98 amplicons were needed in the Arctic style approach. Now at Oxford Nanopore and with the scientific community, we also released a kit called the Midnight Kit. So this is used for viral whole genome sequencing where we use longer amplicons. You can see that they're 1,200 base pairs, hence the name Midnight. And with that, there are fewer amplicons that are needed to span the entirety of that viral genome. So only 29 in comparison to the Arctic strategy. And you can see, for example, this has been used for other viruses such as HIV or Hep C. And it's quite an interesting approach because if your samples are high quality and you can do long amplicon generation, then you can just sequence effectively the entirety of that viral genome. For example, you see here the Hep C genome is around 9 kb, and the authors generated one very long amplicon to sequence the entire genome. So again, that's quite a tunable approach where you can generate PCR data and run it on nanopore sequencing. So low cost, but it also alleviates you from having limited short amplicons. We have a fully kitted solution that again offered through our partners at Avantor that has all of the reagents bundled together. So this, the primers that are included are for SARS-CoV-2, but we also see the community really, that you can get this kit and just add your own primers in if you're interested in tuning those to different viral genomes. So again, an all-in-one solution, fully kitted, and only one order needs to be placed. Okay, and in the last few slides, we're just going to talk about an exciting, change in the way we can look at samples, which is pathogen agnostic metagenomics. So we've talked about, isolating microbial samples. We've talked about targeted approaches like 16S sequencing or targeted viral whole genome sequencing. But what if you don't know what's in that sample to start with? This is an exciting area for nanopore sequencing because, again, labs that have infrastructure or are interested in getting started can really have a low-cost screening tool for looking at anything, in theory, what's in that sample. So instead of asking the question of, what is in the sample with a targeted approach, we can start to look in an agnostic fashion and pick out on all of the DNA and RNA within that sample to make gain some inference into any microbes or possible pathogens, as well as antimicrobial resistance markers within that sample. Beyond the ability to detect anything, again, having long DNA fragments that we can pull out of that with nanopore sequencing is quite impactful for MAGs, or metagenome assembled genomes. So we see an example here, which is benchmarked on a common standard called the Zymo community. And so what we see is if these are sequenced with short reads, it's very difficult to assemble those metagenome assembled genomes. In part because, again, there are repetitive elements across these various genomes and maybe they're mobile elements. And if the reads are short, then it's quite difficult to piece that puzzle together. Conversely, with nanopore sequencing, we can assemble these really high quality and closed metagenome assembled genomes, which is quite exciting because, again, it's a fast workflow, but with long read data out provides very compelling advantages over short read approaches. Here we see an example, not unlike the 16S workflow. This is the workflow metagenomics. So again, after you have base called your data onto your local laptop or system or computer, you can just upload your base called FASTQ files. And again, this can do mapping to databases and gain inference again on the microbes that are contained within that sample. You can point to public databases such as NCBI or your own custom databases if you have more of a curated database that you like to look at with your data. And then again, intuitive reports where we look at the classification and all the lineages detected in that sample. So let's end just before we take some questions on a really interesting and exciting case study in this space. So here we're looking at respiratory metagenomics. This is was a translational study conducted in the UK. And so what they did is they effectively ran this as a pilot service. So if samples were submitted, these could be BAL and tracheal samples, they would effectively get the sample and split it. And so they would do a nanopore sequencing based approach to look at anything in the sample, and then routine microbiology testing just to verify what they had picked up with classical techniques. So I think the exciting thing to call out here is if you direct your attention to the bottom right-hand side of the chart, you can really see how you can stagger the amount of information as it's coming off of the instrument. So really within about 30 minutes or so, you can get information on the microbes that are detected. You can run the sequencer for a longer period of time to pick up more antimicrobial resistance markers. And then you can run the sequencer for, say, 16 hours to get whole genome assemblies from that sample. So it's quite flexible, and I hope this, sparks some interest because you can think about, not a fixed runtime, but how much data do you need to accumulate to answer the question. So if you want to get quick detection, that can be done in a quite short amount of time, or you can run the sequencer for longer periods of time as those reports are coming off to generate more insight into the genome sequencing of the sample. So all in all, this on average with the workflows for their samples were completed from receipt of sample to generating these reports in about 6.7 hours of turnaround time. So if you compare that to sort of classical microbiology and culture of around 40 hours, I think this is a really exciting time for implementation and assessment of metagenomics for these kind of applications. So with that, I will stop screen sharing in a moment and just want to say thank you so much for joining us today. We did want to leave some time to take questions. So I will go ahead and stop my screen share and we will move on over with my colleague Frederic to take some Q&A.
Yeah, so let's get started. Thank you, Aaron, for this great presentation. So first question that we can take, let me see, there's quite some in there. So how often and how usable is this for use in plant genomics sequencing? So to give an answer on that, actually there's a lot done on plant sequencing, depending a little bit on what you're doing. So of course, you're more in the seed part. A lot of de novo assembly can be done, but also targeted sequencing and virus looking at infectious implant as well. So yes, it's often used, and we have quite some articles on this. So please contact your rep, and then can give you some extra information on that. Hope this answers your question.
I see a question here from Marco. It's asking, why do you recommend the rapid kit for bacterial whole genome sequencing and not the ligation kit instead? So that's a really good question. I would say overall, we provide guidance on kits, but they're really interchangeable. So you could use the ligation kit or the rapid barcoding kit. As Frederica mentioned, some of the attributes for the kit might be more or less amenable to what you're trying to do. So I'll give you an example here. You can use the ligation kit for bacterial whole genome sequencing, not a problem. But we found that the rapid kit is faster and it's lower cost. So you don't need you don't need third-party reagents with a rapid barcoding kit. And there's another feature that's actually quite interesting because if you extract the DNA from that sample, but some of your plasmids are still circular, they haven't been effectively broken and linearized, the rapid barcoding kit will cleave that molecule and you'll recover more plasmid DNA as well. So that's been shown, and again, so I think from our recommendation, it's faster, it's lower cost, and can improve some features of plasmid and bacterial recovery. It's not to say you can't use the ligation kit though, that's a great kit as well. So that's some guidance there.
Thanks. So next question is, how do you call methylation, and do you need to perform bisulfite sequencing? Well, no, everything is in one assay. So basically what we're doing is when we're reading your DNA, if your DNA is methylated, it will give a different signal. So these different signals can be just used, and we will do that in our base calling. So you don't have to do any extra assay for this. Hopefully it helps you.
I see a question here about DNA extraction kits. So the question is, are you developing DNA extraction kits? Is there a specific one you recommend for long read sequencing? So that's a really good question. It's really something not to be overlooked because, as you saw, nanopore sequencing is quite agnostic to the DNA that you present to the nanopores during the library preparation. So what I mean by that is you want a kit or an approach. If your goal is to generate long sequencing reads, then you need to generate long DNA fragments to present to it with a kit or a protocol. So there's not one single kit. At Oxford Nanopore, we don't provide DNA extraction kits, which is in part why you should definitely discuss with your local Avantor rep, because we have guidelines and many kits that they offer that produce excellent and high quality DNA fragments. So I would say that's a great opportunity to discuss with them, and we can provide some links and resources as well.
Good, thanks. Let's see, there's quite some questions. This one. Can you explain the difference between ligation and a rapid sequencing kit? I'm not sure what is the best for me. So if you're not sure what is the best for you, please also talk to your account manager, since it really depends on what you are doing. So basically, ligation kit will ligate your motor enzyme to your DNA. So then you have influence on the length of your DNA. With the rapid kit, because we're using transposome, it will cut your DNA a little bit, and that will give you less possibilities there. So it's depending on your question, the amount of DNA, and therefore we already showed you the slide with all the information on the different library kits, and this can really help you to decide which is the best. So please talk to your account manager, and hopefully they can help you to guide you to the right kit.
Great, thank you. I see a question here from Sergio. So the question is regarding Minnow. The workflow allows us to go. Does the workflow allow us to go into taxonomic classification? Is it possible to perform the same analysis with an amplicon approach? So yes. Just as a bit of an orientation, the software called Minnow is what you download to perform sequencing. So for example, let's say you have a laptop, you download Minnow and you can plug it in, and that allows you to really start the sequencing run. And do the settings for sequencing. Now, if you want to analyze your data, that's where we have our Epitome pipeline. So that is a software tool, again, that can be downloaded to your computer, or it can be run on the cloud. And that would enable you to do those analyses once you have base called that data. So yes, you can use either 16S or ITS and generate those types of reports and tabular outputs, where you can do some of those downstream kind of typing analyses as well.
So let me take this question. How long does nanopore sequencing take? So it's really depending on you, because you can decide how long it will take. So since it's real-time sequencing, you can get your first data out within a few seconds and already analyze that. So if you have enough data, you can stop your run. And if you're using this, the MinION flow cell, you can actually reuse that flow cell for your next one. So the total use of a MinION flow cell is 72 hours. So basically, if you have pools, you can still use it. Do wash your flow cell after each run, and you can probably use it multiple times.
Great. So I see a question here from Fernando, which asks, what is the kit for metagenomic analysis? How much time does it take for the analysis? A little bit kind of like how you addressed that question, Frederica, on the time. It's a really good question. It would be important to understand your sample type and the question you're trying to address. So I say that because there might be different kit recommendations if you're working with soil or water samples. That might be quite a complex community. There are several commercially available kits that we recommend that work quite well, and we can provide links and the emails to those direct kits. And especially, again, good to discuss with your Avantor rep on their offerings. You can get those all bundled together. So lots of good offerings, but we would just probably want to verify, is it, the sample type, is this, a specimen where you want to look at something that has a lot of host background. That's an important consideration if you're looking at human samples. So for metagenomics, if it's a human sample, of course, a lot of your proportion of DNA is going to be from the human host. But if you're interested in microbial samples, you want to try to eliminate as much of that host background as possible. So these are a couple scenarios where we have protocols on our webpage for guidance on how to eliminate host background. This can be, for example, differential centrifugation and saponin treatments. These remove a lot of host reads. So it's a little bit of a nuanced way to address your question, but metagenomics is a broad field, so we would probably want a little more information. But that is all to say that we have lots of really great resources on our webpage and with our Avantor partners. The amount of time it takes, as we saw, can also depend, where really the fastest way to a question for metagenomics is if you're just trying to identify the taxonomic profiles. That's a pretty quick approach. We see even in complex communities that can be done, in minutes to a few hours. If you want to get whole genome assemblies out of that, we'd probably recommend you run it for longer periods of time, because then you'd get more coverage to generate metagenome assembled genomes. So again, a little bit of a nuance, but you have a lot of flexibility for what you want to try to address with metagenomic sequencing. So hopefully that helped to answer that a little bit.
So we have another question from Marco about accuracy. This is, of course, always a question we get, so I would love to answer this one too. So accuracy is one of the things that is often said, that we are not accurate enough. And I want to ask a different question on that, because basically we improved our technology a lot in the last coming years. And as you saw in the presentation as well, we are now getting to a Q20 easily, meaning that we're over 99.9%. This gives you actually all the information that you need. And if you look at what you're now doing with short reads, actually we can, because we get those longer reads, we can get much more information out of that. And in the end, that is the most important part, that you get the information that you need. So I hope this will answer your question, Marco.
Yeah, and I'm happy to build off that, Frederic, which is, several of the workflows we talked about today, especially if they're, these kind of sample types, you're not generating one read, right? You're generating, millions of reads. And so oftentimes, it's, I think, important to think about the question you're trying to address as well, which is, if you want the highest accuracy, generally you can create a consensus. And so with that, it's as a single nanopore passes through, as a single molecule passes through the nanopore, that's our Q20 accuracy. But with reasonable coverage, say like 10 to 20 fold coverage, you're looking at Q50, Q60 consensus scores as well. So depends a little bit on the question, but I think it's important just to take that into consideration, where you have raw reads and you have consensus reads. I see a really nice question from, I think, Michela. So the question here is, we're cloning our therapeutic genes in plasmids and normally do Sanger sequencing to check if our cloning attempts worked out. I always thought of nanopore as a multiplexing thing, but whole plasmid sequencing had me listening up. What would be needed for steps or kits for whole plasmid sequencing, and how much would that cost per reaction? So that's a really, really nice question. Of course, plasmids are small, generally speaking, maybe let's say 10 kb for a thought experiment. And really within minutes of sequencing runtime, you would have enough data for a plasmid on nanopore. So that's generally why users, if they have it in their own lab, they may want to multiplex, because there's lots of capacity either on a fungal or a mini flow cell for plasmid sequencing. So you could do, let's say, 12 to 96 samples easily. And so the more samples, of course, you sequence on a flow cell, the lower the cost per sample. So just to give you a ballpark estimate, if you ran 96 plasmids on a mini flow cell, you would be less than $10 a sample. It would effectively be approaching Sanger level costs. And of course, if you run those on a fungal flow cell, which are those lower cost single-use flow cells, that can easily be done with plasmid sequencing as well. You can either run fewer samples or even probably up to 96, which puts you in sort of a dollar range for a plasmid sample. So again, that would be really just our rapid barcoding kit, which you can get through Avantor. And then we have a workflow, again, where you could use plasmid preps of your choice, rapid barcoding kit, and then sequencing and analysis. So a few ways you could do it. There's, of course, many service providers offering this as well. So you have a lot of options for doing whole plasmid verification, so you don't miss anything that you might miss here.
Thanks. So another question is, so I mentioned that the gridiron will come soon via Avantor. So we don't have a timeline exactly, but if you have questions on that, please already go and talk to your Avantor rep to see if this is an interesting device that could help you in your research. So the next part of this question is, what are the advantages of a gridiron over the MinION device? So MinION device And I'm now talking about the MinION NAC1B, so just the small one has no computer in there, but you have to have your laptop as well. So the GridION device has 5 possibilities to load flow cells, and you can use them separately. So meaning that you get really high throughput to your lab, and it includes a really strong computer. So we're talked about adaptive sampling, so this will be ideal for a GridION application. So if you're really going from a small lab to a high throughput lab, you just get more possibilities to do more sequencing. And do talk to your rep if you have an interest for the GridION.
Great. I see a question here from Xavier. So the question is, what are the drawbacks of the Flongo flow cells or the MinION flow cells for metagenomics? So again, a really good question. It all comes down to throughput. So again, as we heard, the Flongo flow cells are really sort of those single-use type, low-cost flow cells, producing in that range of a gigabase, again, depending on DNA quality a little bit. A MinION flow cell is going to put you closer to like 15 gigabases to 20 gigabases, again, depending on the sample input quality. So that's really the question there you would ask for: how much coverage do you expect you would need, or sensitivity that you would need for metagenomics? That depends a little bit on the complexity. Again, I would, just to give maybe my personal opinion here, if you were doing like soil metagenomics, there could be thousands of microbes. That would be a better fit for a MinION flow cell because you'd have better sensitivity, especially for less abundant organisms in the sample. If you're dealing with something that you expect a reasonably small number of microorganisms in that sample, the fungal might be great for detection of a simple community. So again, there's nuance for metagenomics, but that would be, I think, a starting point for how you're trying to address that, how much coverage that you need. And I'll just add one more point where I think if users are just getting started out and you want to really systematically address that question, I would recommend you run a pilot where you generate more data than you need, and then you can actually subsample that data and see if you can answer the same question. That gets you down to a level where you can start to say, for my experimental design, I really only need 2 gigabases of data or 1 gigabase of data because my, species assignments have been plateauing after that amount of data. So a little bit of nuance to the question, but hopefully that helps get you started.
So one more question on sequencing eukaryote whole genomes, especially when they're larger genomes. So yes, this can be done. So if you're looking at the 3 gigs genome, you will get a coverage, a low coverage with a MinION flow cell. But within Oxford Nanopore, we also have the PromethION flow cell, and that probably would be a better solution. Unfortunately, these are not sold via Avantor, but there are options to do this if you want. But first of all, you, of course, can test this on a MinION to just see if this already is answering your question. Be aware, you will not get more than a 5 to 10x coverage.
Great. I see from Anna, she mentions she's interested in the possibility of applying the technology for identification of protozoa and mollusks. So again, that's great. There's a lot of work out there being done with nanopore for identification, again, using your target of interest. So if you're looking at, let's say, 18S or some kind of marker that you're interested in protozoa, you can use that for low-cost rapid amplicon sequencing with nanopore. So there are a lot of barcoding style projects for species diversity estimates, for taxonomic assignments, for looking at, again, microbiomes. All of these are often targeted approaches. So effectively, if you generate your marker of interest, you can use nanopore sequencing for high throughput amplicon sequencing. So it's a very versatile system for that. So if you have any follow-up questions, please do feel free to reach out, but we can pass along quite a lot of resources for that.
Thanks. One more question, because we're running out of time, I think. ONT is applicable for recombinant sequencing of antibodies. So basically, we can sequence anything. So do talk to us about this, and I'm sure this can help. Sorry, we don't have much time anymore. So Maria, I think I'm giving it back to you.
Great.
Thank you, Arun and Frederique, for this very interesting presentation. Now we will close here the session, and I would like to especially thank you all, participants and speakers, for your interest and collaboration in this webinar, and hope you have enjoyed and found it useful. Questions that have not been answered here, they will be answered directly by email. No worries on this. Please do not hesitate to contact your Avantor Life Science specialist if you need more information and support about this topic. And said that, thank you and have a nice day.
Thank you.
Bye-bye.